Thank you very much, Danielle, and to the consortium for the invitation to talk here. It's very, very exciting. Can everybody hear me properly? Is that fine? Yeah, good, okay. So I'm going to talk about a project, most of which was indeed my own work. It's a kind of toy fun project that I've been doing for the last few years, and it's all unpublished, but really, you know, I don't mind recording it, and I hope to get this out one day when I have time, but it's really just a lot of fun, I think. So, yeah, the previous talk from Irene showed us very, very well that we cannot simply look at sequence and see it as a bunch of text. It's doing more than that. DNA is more than text, and other molecules are involved in bringing this molecule to life. And one particular kind of DNA that I became really interested in many, many years ago is the DNA that we call zDNA. It's a left-handed helix, differently from the right-handed helix of bDNA that we know so well. It twists to the left. And the name zDNA comes because it has this zigzag shape along the backbone. So the orientation of the helix is flipped. Why does it do that? When it does do that, you can see that the bases are not lying nice and flat like they do in bDNA. And that's actually the secret to why zDNA forms like that. It's formed at alternating stretches, specific sequences, mainly alternating purine-pyrimidine bases, unlike these triplets, which are purine-rich. These are alternating. And the steric hindrance between pairs of bases, which are purines and pyrimidines, means they're more comfortable actually flipping away from each other, and that flips the entire backbone over to a left-handed structure. Now, over many, many years, it's become clear that zDNA does exist in vivo, and it does have many roles. And the important thing about it, I think, is that it helps to unwind DNA. If we twist DNA to the left, we're actually opening it. And that's really fundamental to its function. And indeed, DNA, which is wound around nucleosomes, and also DNA that exists in bacterial chromosomes, is twisted to the left. It's constrained as negative supercoiling contained in those nucleosomes. And any process that removes nucleosomes is going to leave behind local unconstrained negative supercoiling. And this happens during many important biological processes. Now, this negative supercoiling then favors the twist to the left, which is happening with zDNA. So if you have this negative opening supercoiling, that will eventually cause certain sequences to flip over to the left. And that will only happen when they're negatively supercoiled. And when they do that, that zDNA actually absorbs those negative supercoils. So it's both a sensor and a modulator of negative supercoil. And I'll show you more about that in a minute. There are in various species zDNA binding proteins that can recognize and stabilize zDNA. I'm going to show you a movie so you can really visualize this. Here we have a piece of DNA, and the red part represents any Z-forming sequence that can form zDNA. The yellow part cannot. It's always going to be bDNA. And now when we untwist this, you see eventually upon opening the bDNA, it will form a supercoil. There's the supercoil. Now if we let the zDNA do its thing, the zDNA sucks up that supercoil. You see this in slow motion here. The bDNA is no longer supercoiled. The zDNA carries it all. And so zDNA absorbs negative supercoiling, but it needs them to form, and bDNA gives those supercoils over to zDNA. You know, what this tells us, I think, if we do not consider these very, very dynamic structural transitions, we cannot fully understand this DNA sequence. And a lot of the work in the field of zDNA has been trying to predict which sequences will form zDNA and in what way. So there's also a dark side to zDNA. There are various publications that show, in fact, that zDNA can cause genomic instability. Z sequences are enriched at mutation hotspots. zDNA is mutagenic, and there are DNA repair proteins, in fact, that bind zDNA and introduce breaks. Why does that happen? It's because when you have the junction between B and zDNA, two bases actually poke out and stick out from the helix, and they're the fragile ones. That's where the breakpoints occur. So zDNA has a dark side. But that particular fragility also has a very interesting role in evolution. Here's an example where a zDNA forming sequence is very enriched in an enhancer, which makes this pelvic fin in the fish. And those sequences are fragile, and fish that lose that enhancer, they're perfectly okay, but they've completely lost that structure. So zDNA can even drive evolution. And what I want to talk to you about today is my favorite topic. I haven't got an answer to this, but this is what I'm going to explore today in this work, whether zDNA could also be an epigenetic switch. Now, our favorite regulatory elements, we call them memory elements, they regulate genes. A memory element can register what's happening at the gene. It can perpetuate that through DNA replication and mitosis, and it can then signal back to the gene after those disruptions to remember what it should be doing. Many years ago, we discovered, together with Mark Rimsmeyer, that sequences with Z-forming potential are enriched in our favorite class of epigenetic memory elements. You know, and this, for me, this is just a very compelling idea that one piece of DNA, we know that one piece of DNA can be both an activator and a silencer. Is part of that switch actually a structural switch? So this idea started, as I said, many years ago, and I've now had time to do a little bit of work on this. Our favorite elements are called polycomb trithorax response elements, or PRE-TREs, and they contain zDNA. I'll show you the evidence for that later on today. They bind two types of proteins, silencing and activating proteins, which work antagonistically on those elements. And as I said, the same piece of DNA can activate or silence the gene depending what binds to it and what information it gets from the gene. And those elements can then maintain the memory of expression states. Now, one kind of very long-standing mystery in the field is that the proteins are very conserved between flies and vertebrates, but the sequences of those elements are not. And so, as I say, we found that they do contain these Z predictions, and we wonder whether that's part of the puzzle, that these zDNAs may play a role actually in the function of the elements, and that's what we're going to be investigating today. My talk is divided into 4 parts. I'm going to start off with a bit of historical stuff in silico. How do we actually find these sequences in genomes, and what do they look like? There is a very beautiful paper from 1992 about an algorithm called zHunt2, and it uses statistical mechanics to evaluate for every pair of bases what is the likelihood that they're going to flip out to this Z conformation. You can then, into that algorithm, give any sequence and you'll get a score predicting how likely is it to make zDNA for that score. Now, here's a plot of different predictions from that paper. And I want to point out here that what we're doing is we're asking how much negative supercoiling does a sequence need to flip over to the Z form. And I've plotted this as more supercoiling at the bottom and less at the top. And what you see is that the lower the Z score, here are the low Z scores, the more supercoiling it needs to flip, and the high Z scores are more able to flip with less supercoiling. And that's a very nice number to know. Units of Z score here, KB, it's just an indication of how much sequence would you have to search, how much random sequence would you have to search to get a Z score that's equally good at flipping. Now, obviously, that's very nice, but we'd like to know whether those supercoiling densities are actually realistic in vivo. And so you can, supercoiling density in bacteria has been measured. So in fact, bacteria have very, very high negative supercoiling, and it would be sufficient. Here's the more supercoiling. All of these sequences would flip in bacteria, no problem at all. It's been much more difficult to measure in eukaryotes. I did this little back of an envelope calculation based on what we know about the density of nucleosomes and how many negative supercoils they contain. And depending how you calculate that, these sequences might have trouble flipping. All of these would be okay. So if we find Z scores in vivo, which are this high, they would flip theoretically, and these ones might need help. So, but it is realistic to expect that the densities, the supercoiling densities, are sufficient. These are the sequences, and what you see immediately, these sequences were theoretically calculated in that paper. You see immediately that the GT repeats have very low Z scores, And the same length of a GC repeat is way more powerful. It's orders of magnitude higher in a Z-score because of the shape of those bases. And of course, what we would like to know, these are theoretical examples, what kind of sequences and Z-scores actually exist in real genomes. And very interesting, actually GT repeats are much more common in genomes than GC repeats. Here's just the example from human and Drosophila. We have a lot of GT repeats. GC repeats are rare. And incidentally, the rarity of GC repeats in vertebrates has been attributed to DNA methylation, but that cannot explain their rarity in Drosophila because Drosophila don't have DNA methylation. So, you know, maybe they're just like super Z-forming sequences and they mutate and they disappear. Could be. So that's interesting. Where are the GT repeats then that we have a lot of? Where are they clustered? The GT motif was actually originally first identified by Mark Grimsmeyer and myself by motif discovery in Drosophila polycomb response elements. We found that it was enriched there. Later work has identified a protein that binds it on the DNA level and a co-localization with polycomb group protein. So that's definitely doing something at the DNA sequence level. When we look into vertebrate mouse polycomb binding sites, this work was done by members of my lab recently. We find that GT repeats are also enriched in polycomb binding sites. And if we just look here, what I'm showing you is very short repeats going up to very long consecutive repeats across the board. And what you see is that the short repeats and the longer repeats are more enriched in Jared2 and SUSY12 binding sites. And if we calculate the Z-score for each of those, we have a reasonably healthy Z-score for repeat of that size. So it also holds genome-wide. We can then also apply the same question and look at GC repeats. I told you that they're rare genome-wide, but we actually find huge numbers of them in polycomb binding sites. So they are enriched there. And again, in the literature, this prevalence of GC-rich regions in polycomb binding sites has been attributed to CPG islands being the main recruiter of polycomb group proteins. That is unproven, and I would like to propose today that there might be more to it. These GC-rich sequences might be contributing to zDNA. Is that true in real polycomb target genes? I've just told you about repeats. How about the real sequences? Here is an example of our favorite fly locus and our favorite mouse locus, which is the homologous gene in the mouse. That's the vestigial locus in the fly, vestigial like 2 in the mouse. And you can see there are very high Z-scores across both loci. And the red part is our favorite regulatory element, that's the PRE-TRE, where we find clusters of these. Here's a cluster there, and in the mouse, we have a cluster of very high scores. If we look at the real sequences, it's quite interesting. The sequences themselves, yes, they do indeed contain these long CA or GT repeats, but the really most powerful ones, the ones with the highest score here and here, that's B here, this one, and D in the mouse. They're actually rather cryptic. They're not just these repetitive things, but they have this high Z-score because of this consecutive properties of the nucleotide pairs. So, you know, that's very interesting and worth looking into further. So what I've shown you to summarize this part is that zDNA sequences are predicted by this algorithm. We find them clustering in these elements. And so obviously we want to know what they're doing. And before I tell you that, I am going to take you on quite a long detour around how we actually do this, because you know it's very important to have good assay. And what we're going to do first is think about how we actually analyze this in vitro and can we improve existing assays. So we're working in vitro then with circular plasmids, and we have circular plasmids which are identical except they just have 28 base pairs different. So here's one that doesn't make zDNA. It's got 14 times AT. This one can make zDNA. And as I showed you in the video, when the plasmid is relaxed, the zDNA will not form. It will be bDNA. But as soon as we coil it up, it's going to start flipping into the zDNA. How do we see that? We see that with a 2D gel. Each of the spots in this array is a ladder, is one rung of a ladder of topoisomers differing by only one negative supercoil. So we can really quantify single differences in supercoiling. And you can see that the plasmid that has zDNA already looks really different to the one that doesn't. And I'll walk you through that, why that happens. So we have a first dimension here, and we run a second dimension in that direction. And when we come to the very highly supercoiled forms, remember from the video, the zDNA will form when we have high negative supercoiling. But as soon as it does that, the zDNA absorbs negative supercoils, and we lose supercoils from the bDNA part. We just have a Z part. And the big difference in topology here means that this form migrates more slowly. And this is why it's higher up in the first dimension of the gel. So this shift reflects this large change in topology caused by the zDNA. So we can, from this, tell at which supercoiling density exactly does that shift start. And we then looked in more detail what determines the pattern of those spots. And I'm going to take you through this. In the first dimension, it's just how much Z score we have and whether or not we have Z stabilizing agents, which causes that shift. And that determines the position of that shift. How big is that shift? This also gives us information. It depends on how many supercoils are absorbed, and that is quantitatively dependent on the length of the Z forming sequence. So here we're actually absorbing 5 supercoils. In the second dimension, everything changes. The second dimension is purely analytical. There's no more zDNA. We're just trying to resolve the spots that were shifted in the first dimension. This depends entirely on the concentration of an intercalator called chloroquine. If you're interested in the theory, we now have the most relaxed part at the left of the gel, supercoiled on the right, and chloroquine is an intercalator, and it untwists DNA, which results in a compensation to 2 positive supercoiling. And this is a quantitative relationship, but surprisingly, we don't know much in a very systematic way about those parameters. Some of them have been studied theoretically, but experimentally, nothing has been studied systematically. And there's a very good reason for that, and that's because the experimental protocols for running these gels are extremely slow. And so what I set out to do a couple of years ago is see whether I could speed that up a bit and get more out of it. So can we accelerate this assay? And to cut a long story short, yes, we can. I use a mini gel, a little mini DNA gel, and we then, by doing that, we save enormous amounts of time and buffer, and we end up with being able to process 10 times more samples than standard methods. And, you know, that doesn't mean we can go home early. It means we can do more. We can test multiple conditions. We can run several hundred gels in a space of a few weeks and really systematically test conditions because we have a fast turnaround. So, you know, we can just run stuff through it and get more information. So what we've been able to do then is actually experimentally, systematically test every single one of those things very, very thoroughly. And I'm just giving you an example here of increasing the chloroquine concentration in the second dimension. This looks pretty, but it also has an interesting, you know, there's an interest in doing this, and that is to find the condition where we catch that shift. Here we can't see it because it's buried in the corner there, but we start seeing it here and here and here. So that's why it's interesting to do that. It doesn't tell us much about biology, but it helps us to determine when we can see that zDNA forming. Okay, so I've shown you that we have a faster assay, and that means we can comprehensively explore all of the parameters experimentally. And that enabled us to then go further with theoretical work. There are decades of existing theory on zDNA, and to get our heads around this, I developed a program called the Simulator. And the Simulator does nothing more than simulate 2D gels for us. But the important thing is that it is based on topology theory. It incorporates Z-score, gel conditions, and all the chemicals that we put into the gels. And as you can see, here's the experiment, here's the theory, it looks good. And I'm claiming that it quantitatively predicts the 2D gel shape. And just to prove that to you, here are the numbers of the topoisomers, and every single one simulated by the simulator is in exactly the same position as it is on the gel. Here's just an example of what you can do with it. The user gives inputs. You can give any size of plasmid, any Z-score, and so on. Different chemical conditions. It will predict gel patterns for a whole range of experiments. meaning you can select, you can still do 10 times more in the lab, but you can then also select before you do your experiment what conditions you're going to use, so you waste even less time. Yeah, so as I say, that's the simulator. How well does that really predict real data? I'm just going to flash this in front of you to show that, we can have gels and gels simulated, and they really look very similar. So it does a good job because it's fitted to the experimental data, right? So there's perhaps no big surprise there. But what becomes interesting then is, can we now use that to fill in the gaps in the theory? Because not everything's been done theoretically. Remember our gaps, we have different things that are not completely studied. I'm going to go through this a little bit quickly in the interests of time. But if you're interested in the theory, I'll be happy to talk more to you about that later on during the questions. So the first thing I said, there is a relationship between Z-score and supercoiling density, which had been studied theoretically, not experimentally. We've now defined that experimentally, meaning that we can interpolate between those missing points and come with a single equation that describes the relationship between supercoiling density and Z-score. That is not trivial, because one used to have to do this by these simulations using the Z-hunt, and it wasn't possible to simply have one analytical solution for that. So that's useful. The next thing we looked at is the effect of stabilizing agents. This hasn't been studied at all theoretically, and we were now again able to mathematically define that relationship. Now, having experimental data points for several concentrations, we can then interpolate and give a prediction for any condition of stabilizing agent. So we provide an equation for that. And finally, the effect of chloroquine. Again, that has not been studied systematically in terms of its relationship to stabilizing agents and what happens in the second dimension. So we've been able to do that as well. Again, we have certain experimental data points, and we interpolate between them to give an equation, which is where X and Y are in terms of also the concentration of stabilizing agents. So we've been able to fill in that and consolidate and extend the theory that exists. So that was just a quick tour through the simulator and the work that we've done to sort of understand and consolidate the theory. And what that means, I think that's the important thing, is that we have an accurate prediction of the behavior in our assay for any Z-forming sequence. So then we can go to the real sequences that we're interested in and ask what they do. And this is where I now come to the final part, which is the in vivo work that we've done. And I told you that we have predicted zDNA in fly and mouse PRET-arrays. And so we can ask now in vitro, do these sequences form zDNA? What are the quantitative parameters of that? And does it actually affect the function of the elements? So just to remind you, we've got these target loci that we're interested in, and they contain several Z predictions. What I did was to clone these little sequences into a vector, which is so that the plasmid is identical except for those sequences. And then I can ask, do those sequences form zDNA in vitro? And now you've seen the assay. What we're asking is, do they make this bump in the shape of the 2D gel? So these are our sequences that are cloned, and you can see in every case here, here's the experiment, here's the prediction, we get the nice bump, here's the control with no bump, here's this one that shifts at -9, here's another one that shifts, this one shifts, this one shifts, and they all shift. It's interesting, they all have different personalities, and that is really interesting. Part of that we can capture with the simulator, which says that our model is quite general. Part of it we can't, which is where it gets really interesting. So if you look at this one, for example, it shifts at -9. We can capture that with the simulator, but it does something very weird at -12. So there's some cooperativity going on there. That's something we would like to explore further. I'm going to now focus on this particular sequence, which is from the fly. It's this sequence G, which is fairly complex, and it does a nice shift at the topoisomer -9. And I'm going to then work further on that and ask, can we now change the Z-forming properties by minimal changes to the DNA sequence? By which I mean, we're going to be looking whether this shift position in the gel, does that change when we change the sequence, and how much of the sequence do we have to change to do that? And here are the point mutations that we're making. We're making minimal changes. We're just changing a GT run to a GC run, and that should be stronger. The prediction is that it has a high Z-score, and this one should be lower. We're taking out those purine pyrimidine repeats completely and just replacing it with a different sequence. And you can see in vitro when we do the experiment, here's our wild type sequence. These are just plotted against the Z-score for the superhelical density. And what you see is that the wild type sequence I showed you shifts at -9. The high Z-score shifts earlier, it shifts at -8. And so it needs less supercoiling, meaning it is indeed easier to make zDNA from that sequence. The low Z-score shifts at -11, it shifts much later, so it needs more supercoiling. It's more difficult to make zDNA. So, what we're doing here is changing 2 or 3 nucleotides, and you see this very detectable change in the 2D gel, which is nice. But, what happens in vivo? We'd like to actually know whether the fly cares about that. So we're going to put them in animals, and we're going to see whether the animal can detect that change in DNA structure. Now, we're looking at the fly locus vestigial again, but we are not, I should warn you, we're not manipulating the element in its endogenous locus. We're making a transgenic assay where we take out this sequence and we hook it up to a reporter gene. So here's the PRE-TRE sequence, it's 1.6 kb. We put it next to a reporter gene, which normally gives the flies a red eye color. Now, generally in transgenic flies, if we take completely different elements here, they will have a personality. They will affect the expression of the reporter gene in a very characteristic way, and it could be low, medium, or high. When we take the wild type sequence of this PRE-TRE, it gives a medium eye color, so it's kind of orange. And then, of course, we can ask that's a good position to be in, because we could go up or down from there. If we change the zDNA sequence and change its zDNA properties, are we going to go up or down? What's going to happen? So we could actually do that experiment. For those fly aficionados in the audience, I just point out that we integrated all of these at identical genomic locations. So there's no confounding effects from surrounding genomic sequence. And this was done by a former PhD student in my lab in Vienna, Helena. So you see how long I've been interested in this. I actually got into this in about 2010, something like that. So it's been a long journey. Right, so here's the construct. This is our PRE-TRE, and I'm just showing you little motifs for different binding sites. The important one is comb gap that has a GT-GT repeat. So you can see this thing is full of GT clusters. And what we're doing is messing around with this one. This is 40 base pairs here, shown it's just here. So we're only changing that. We're changing, again, the same 2 nucleotides for this construct and 3 nucleotides back for that construct. And what we see is that it does indeed have a profound effect on the properties of the element in vivo. We have the wild type, which has its Z-score of 10, and the higher Z-score has a darker eye color, meaning a higher gene expression here. So the gene expression is correlating with a higher Z-score. When we bring it back down to a lower Z-score, that's this one, we restore silencing, but we do not actually restore the original sequence. So this is the quantification of that, and we're just measuring in eye pigment. I just want to summarize that. So the higher Z-score in vitro shifts more easily to the Z form. It correlates in vivo with a higher reporter gene activity, and the lower Z-score restores the reporter activity without restoring the original sequence. So there is something more than just this motif, which is helping this thing to work. So I'm going to finish there, and I'll give you back the summary of what I've shown you. In the in vivo experiments, I've shown you that those sequences, both from the fly and the mouse, do indeed form zDNA in vitro. And I've just shown you a selection of those in an in vivo assay where when we change the Z-score, we actually change the function. So just to summarize everything that I've shown you and finish up, in silico, we predict those sequences and we find them in these polycomb trithorax response elements. In vitro, we did, or mainly I did work to kind of get this thing up to speed so that we could squeeze more out of it and enable really measuring these parameters. And that enabled me to then consolidate in this simulator framework everything that we know about zDNA and predict then what it's going to do. And finally, in vivo, we've shown that Z sequences do seem to affect the function of these elements, these memory elements. So I'm going to hand over then to the discussion, and I'd love to hear your thoughts on this. My thoughts on this at the moment is I don't know if it works really via zDNA, but I like to think about this, that zDNA might repel or attract proteins that like to bind this thing. It might like to bind zDNA, or it might hate to bind zDNA. So that's something we're thinking about. zDNA is favored by transcription and replication. So, is it doing something in sensing topology and sensing or signaling, or even in memory? We don't know yet, but I think it's very interesting. And finally, a question very close to my heart that I've been thinking about for a long time: is it actually zDNA that brings the fly and the mouse together, so that we shouldn't be expecting DNA sequence to be conserved, but perhaps the structures are conserved? And that's something we're going to be looking at in the future. In the end, my take-home message is, if we consider DNA structure, we can better understand DNA sequence. And I want to thank the people who've been involved in this. If you work in my lab, you will eventually bump into this project. So a lot of people have contributed to this. I'll start with Mark, because he actually discovered the GT motif computationally in 2003 in PREs, and he made that movie. So thanks, Mark. Other people did various things where I have acknowledged them in the slides. I want to point out Alex and Jeanette, whose work I didn't have time to show you today, but they've been working on aspects of GT motifs and proteins that bind them. Diego programmed the ZHunt2 algorithm for me, and Helena, back in 2010, made those transgenes when I first started getting interested in this. So I thank you for your attention, and I'll be glad to take questions.
Thanks, Leonie. It was very interesting. So I have a first question regarding your in vitro assay. So how are you sure that what you are seeing is not the denaturation of DNA? Because negative supercoiling can also, as you said, increase the probability of making DNA bubbles, or single stranded bubbles that could also absorb supercoiling very efficiently. Right.
Yeah, thank you for that question. I didn't put the slides in, but we've actually done a very beautiful experiment where I cloned the zDNA binding domain of the Z-alpha zDNA binding domain of the ADIR1 protein from my own genomic DNA. It's no mutations. And I fused it to GFP. And I then did an assay, which is the first assay anyone's done, where you actually pull on supercoiled topoisomers out of that mix. And I can show that the zDNA binding domain of that protein actually preferentially binds those, what we're calling zDNA. So that's actually, perhaps I should put that in the paper, right? Especially if you're going to be our reviewer. I should definitely, because I kind of, that's like, okay, Z-alpha binds zDNA, but it actually proves that the zDNA in the gel is zDNA. And we can run that, what is bound to the Z-alpha, we run that back on a 2D gel, and we get only those 4 spots and not the rest. It's beautiful. So yeah, I think I'll add that actually as an important control. Yeah, thanks for that question.
And maybe the second question is regarding the PRE's sequence. So your last experiment where you make the mutation in the PRE.
Yeah.
Did you check if the motif that you have now, because you perturb a bit the motif, do you have also consequences in I don't know, polycomb target genes or trithorax group protein? No, not polycomb target genes. Polycomb group proteins or trithorax group proteins that have less affinity to this sequence?
Yeah, you're thinking we might have made a new version that has now.
I don't know. Yeah, it's just to be.
We cannot exclude that. And I think the way to do that experiment thoroughly is really make every possible mutation, really scan just those 3 nucleotides and change them to every other possible thing, because you can always make a site that binds something that you hadn't really thought about, and oh, now I'm binding something else. Yeah, so we do need to do that more thoroughly. And I think that what we're going to find is there's always a component of binding via DNA sequence, and there may be a structural component. It's going to be quite difficult to deconvolve those. I think it's an interesting result, but yeah, you're right. We might be looking at having just created something new that binds binds better to something.
I just didn't want to misunderstand something fundamental in your in vivo results. When you mutated the native PRE-TRE sequence, such that you were lowering the Z score,
Yeah,
right?
yeah.
And you basically restored.
Yeah.
So hiring the Z score resulted in loss of function of the
Exactly, loss of silencing.
Exactly.
Yeah.
So wouldn't you then conclude that zDNA is not how PREs function? Because if you make it
right,
more,
if you make it lower, then it doesn't care, and if you make
exactly
it higher, yeah, so that if
you're
you
interfering with it,
it messes something up. Yeah, exactly. Yeah, you're right there. That's a good point. The question would be, I think, if that sequence still does actually make zDNA, right? The one that we introduced, I showed you in the gel that it will make zDNA, but it will make it at a higher supercoiling density. So perhaps we haven't gone far enough. I think you're absolutely right. We can't conclude yet that, it's definitely zDNA. It's sort of compelling results, but need more, need more work. So I think, what we'd have to do there, Maria, is really push it so it cannot make zDNA under any conditions. What we also noticed in those lines is, although the total level of pigment is equivalent in the wild type versus the kind of rescued one, if you like, if you look at the eyes themselves, there's actually more variegation in the lower Z score. So I don't know quite what that means, but it means that in biological terms, it means that the element switches more readily. So there's more very pale and dark cells. And in the wild type, it's more homogeneous. So it might actually need the zDNA to maintain that sort of balance. Yeah. What we'd love to do is be able to manipulate the supercoiling, right? What we're doing is changing the DNA sequence. And it's very, very difficult to interpret. So what we'd like to do is, feed them topoisomerase inhibitors or something like that. And then see, does that actually make a difference? We've done the experiment to the extent that we know how much of the topoisomerase inhibitor they can eat without being killed. We haven't done the experiment where we then have enough get in.
So actually, what do you expect the effect of transcription to be? And whether, would this explain the effect that once in a while rounds of transcription may have on PRE function and targeting to target promoters of polycomb proteins?
Yeah, absolutely. I mean, if we think about transcription, there's this sort of polymerase transcription model where it pushes positive supercoiling ahead of it and negative supercoiling builds up behind it. So if you have a polymerase going in a particular direction, and it leaves negative supercoils behind it, and then you have the zDNA forming sequence there, that's going to flip. That would be my prediction, right? And then after the polymerase is gone, that zDNA should be able to absorb those negative supercoils. So I'm just thinking on my feet here, what would then happen? You would sort of somehow counteract the negative supercoiling caused by polymerase, right? You might also, one could also imagine that it would favor further transcription because it tends to open the double helix.
Right, yeah.
Yeah, so you might actually have I've sort of wanted to model this more in a, in a more kind of, what I mean, in a proper model, not just this simulation of topology, but an actual transcription model.
And also the effect it may have on reassembly of a nucleosome.
Yeah. I think nucleosomes don't like zDNA. I think I haven't sort of looked into this systematically, but I think unlike the triplexes that Irina showed that seem to like nucleosomes, I think zDNA kicks off nucleosomes. I read something somewhere about that. I'm not completely solid on that, but I think so. Yeah, I think it would tend to disfavor reassembly of nucleosomes. I think there is also evidence that zDNA is involved in promoter function. It tends to be involved in activation, helps destabilize nucleosomes. What we're really looking for, the holy grail, is something that binds it in the fly. There's the only thing that's been shown to bind zDNA in the fly is topoisomerase 2. Yeah. No,
but that would be so lovely. Oh,
but if COMGAP doesn't bind it, that would also be kind of interesting, right? If COMGAP binds
so that you can test. It wouldn't be so hard with your transgenes, right?
We're in the lab right now doing that.
Okay.
Yeah, we don't have that data, but we're trying it. We're actually trying to do it in vitro. with purified comb gap zinc fingers.
What I will say is fully in line with the discussion you just had with Guillermo, actually. I would say in more simple word, without even going into molecular detail, that obviously zDNA is promote in a way transcription, goes in the same sense as promoting transcription because of the mechanism you said of the relieving the negative supercode. And in my opinion, that would be enough to explain the phenotype you observe going against PRE and PRE mediated silencing. So it was just
I think it would be very interesting to turn the transgene around and see, because we're proposing that it needs to be, the negative supercoiling happens behind the polymerase. So, what's going on? If there's a lot of stuff we can do. I mean, if I had the resources and the time, we would do a lot of stuff. Thank you.
Bye.
Are there elements or molecules that do stabilize the zDNA?
Yeah, there is this ADAR1 protein, which is a super interesting protein. It's mammalian vertebrate protein, and it's involved in immune response to viruses. It has a zDNA binding domain called Z alpha. It's actually got two, but Z alpha is the one that binds zDNA, and that's the one I cloned out of my DNA. So I'm actually doing that. And it recognizes viruses. I think it recognizes viruses that integrate, so DNA viruses that integrate into the genome. When they replicate, they form zDNA, they have Z-forming sequences. It comes in, it combines zDNA, but it's actually an RNA editing enzyme. So it's got a really interesting pathway where it then messes up the RNA somehow by recognizing zDNA. The same DNA binding domain recognizes zRNA, which is double-stranded RNA with the same sequence properties and twisted in the Z form. So this is like mind-blowingly interesting. We haven't really worked on ADAR1 except to clone the Z alpha domain. There are other zDNA binding proteins in vertebrate genomes, similar Z-type domains. And now the paper that I showed you when I talked about the dark side, I haven't had time to look into that, but those authors also show that DNA repair proteins, there are DNA repair proteins that can recognize zDNA. So that starts to get very interesting, and that seems to have been a new recognition, and I haven't had time to really chase that up, and look through what are they, what are the domains. Now, we haven't found anything by homology searches in Drosophila that would have a zDNA binding domain. So, the question is open.
Great. We have one more question from Rafael Murad. He says, thank you for your talk. zDNA has been linked to neurons. Have you found some related phenotypes in the mutants? Memory. It has been linked, for instance, to memory.
We haven't looked, I must admit, but that is a really great question. Do you have a reference? Can you point me to where the neuron linkage is?
Yeah, thank you. Hi, Leonie again. Could you think of a link with the previous talk? Like, as you mentioned, the DNA binding protein also by zRNA, and we heard about triplex RNA. So there might also be proteins binding that RNA in triplex, right?
Could be, except that the sequences, as Elena, I think, Elena, you said they were purine rich, right? Rather than these alternating.
Yes, the one that seems to be more stable in vivo. Yes. Then, of course, other motifs can occur with other sequences, but in a non-physiological condition. So
but I do think it's worth considering RNA, Geneviève, really. Where I've always got stuck with that is that I don't understand where the supercoiling comes from. There's papers that have crystallized zRNA with the ADAR1 Z alpha domain, and it is definitely there. There's an RNA that forms a Z structure, but how does that, how do we get a supercoiling into it? I'm not quite sure, and I haven't had time to kind of really sit down and think about that. I'll tell you one thing for free, which you can all go and work on this if you want, because I definitely haven't got time. The SARS-CoV-2 genome has one massive zDNA score right in the middle of it. And SARS-CoV-2 doesn't go via DNA at all. There's no DNA involved. So that's very interesting. And if it is doing anything, it's got to be RNA. I haven't done anything about it. I sort of found that and thought, I'm not going to turn my lab into a COVID lab. Keep going with Polycom. But it's out there. That's an interesting observation. Yeah. So definitely RNA. There's an RNA bit to the story. Definitely.